Hello dear students, welcome back to another learning lessons in my Physio-Pulse channels. Today we will discuss about Pentose Phosphate Pathway in a simple, easy to understand way. You will find the clear explanations, diagram as well as important concepts here. Before we begin, if you find the video helpful, please like, share and subscribe to support the channels. Now let's understand about the Pentose Phosphate Pathway. First of all, it involves oxidation of the glucose 6-phosphate molecule to form the 6-phosphogluconic acids, which is in turn converted into the pentose phosphates. It is also called the Hexose Monophosphate Shunt pathway. Why? Because in this pathway, glucose 6-phosphate is directly oxidized without entering the glycolysis. Hence, it is also known as HMP pathway or as well as the direct oxidation pathway. Now, this is the diagram where how glucose 6-phosphate is converted to the pentose phosphate, and that reaction sequence if you see step by step. Number 1, first of all, 6 molecule of glucose 6-phosphate in presence of coenzyme NADP are converted or oxidized into the 6 molecule of 6-phosphogluconolactone by the enzyme of glucose 6-phosphate dehydrogenase. 6 molecules of NADP are reduced in these reactions and which is reversible. So this is the glucose 6-phosphate molecule in presence of 6 NADP, and with the help of this enzyme glucose 6-phosphate dehydrogenase, it will form the 6-phosphogluconolactone. In the reaction number 2, what happened? 6-phosphogluconolactone, these molecules is hydrolyzed by the enzyme of lactonase to produce 6 molecules of 6-phosphogluconic acids, and here the enzyme is lactonase. Then 6 molecule of water is also added here to form these 6-phosphogluconic acids. Now in the reaction number 3, these 6-phosphogluconic acids is oxidatively decarboxylated. That means carbon dioxide was removed from these reactions by the enzyme 6-phosphogluconic acids dehydrogenase. Now these 6 molecules of NADP are reduced here, and 6 molecules of CO2 are released, and 6 molecules of Ribose-5-Phosphates are produced. So from this reaction 6-phosphogluconic acids, it will produce Ribose-5-Phosphates molecule. So this is the same reaction sequence here. 6 with the help of this enzyme and NADP cofactors, it will produce Ribose-5-Phosphate molecules. Now 6 molecules of Ribose-5-Phosphate isomerase into the 4 molecule of xylulose 5-phosphate and 2 molecules of Ribose-5-Phosphate in presence of ribulose phosphates 3-epimerase and pentose phosphate isomerase respectively. So what happened if you see these reactions, ribulose 5-phosphates in presence of these enzymes, it will convert to form the xylulose 5-phosphate molecules and ribose 5-phosphate molecules. So in this way it can produce xylulose and ribose 5-phosphate molecules. Now 2 molecules of xylulose 5-phosphate and 2 molecules of ribose 5-phosphate combined in presence of transketolase to form the 2 molecules of pseudohaptulose 7-phosphates and 2 molecules of 3-phosphoglyceraldehyde. That means this 5 plus 5 10 carbon, it will convert to form the 7 plus 3, that is 10 carbon, with the help of transketolase enzyme. And again reaction number 6, 2 molecules of pseudohaptulose 7-phosphate and 2 molecules of 3-phosphoglyceraldehyde combined in presence of transaldolase enzyme to form the 2 molecules of fructose 6-phosphate and 2 molecules of erythrose 4-phosphate. That means once again if you see this 7 carbon and 3 carbon, total 10 carbon, it will convert to form the 6 carbon of fructose 6-phosphates and 4 carbon of erythrose 4-phosphate. Now reaction 7, this erythrose 4-phosphate react with the remaining 2 molecules of xylulose 5-phosphates in presence of transketolase to form the 2 molecule of fructose 6-phosphate and 2 molecule of 3-phosphoglyceraldehyde. So this is erythrose 4-phosphate and xylulose 5-phosphate. It will convert to form the 6 molecules 6-phosphate of fructose and phosphoglyceraldehyde. It will produce with the help of transketolase enzyme. Now in the reaction number 8, 1 molecule of 3-phosphoglyceraldehyde isomerizes into the dihydroxyacetone phosphate, and the enzyme is phosphotriose isomerase. And ultimately the remaining 1 molecule of 3-phosphoglyceraldehyde unites to dihydroxyacetone phosphates in presence of aldolase to form 1 molecule of fructose 1,6 bisphosphate, and the latter in presence of forms fructose 6-phosphate it will produce. So in reaction number 10, 5 molecules of fructose 6-phosphate produced in reaction 679 and isomerized into the 5 molecules of glucose 6-phosphate in presence of phosphohexose isomerase. So this is the reaction fructose 6-phosphate, 5 molecules of this with the help of this enzyme phosphohexose isomerase, it will produce glucose 6-phosphates. So if we get the summary of this reaction sequence, that 6 molecules of glucose 6-phosphate which enter into the pathway after oxidation produce 6 molecules of carbon dioxide in the reaction number 3, and carbon dioxide combines to form carbon number of 1 of glucose molecules, and 12 molecules of reduced coenzyme NADPH reaction 1 and 3, and while 5 molecules of glucose 6-phosphate are regenerated in the reaction number 10. So totally if you see 6 glucose 6-phosphate molecule plus 12 NADP plus, it will produce 5 glucose 6-phosphate 12 NADPH plus 6 CO2. So in other words, 1 molecule of glucose 6-phosphate after oxidation produces 6 molecules of carbon dioxide and 12 NADPH molecules. All the enzymes present in the pentose phosphate pathway are present into the cytosol. So this is all over exactly in pentose phosphate pathway. And if you have to at a glance see the reaction sequence or reactions summary, then you will find glucose 6-phosphate to 6-phosphogluconolactone, then it will produce 6-phosphogluconic acids in presence of this enzyme, then it will produce ribulose 5-phosphate with these enzymes, and here 1 molecule of carbon dioxide is reduced, then ribulose 5-phosphate it will convert to form the ribose-5-phosphate and xylulose 5-phosphate, and this ribose with the help of transcriptolase enzyme it will produce pseudoheptylose 7-phosphates and 3-phosphoglyceraldehyde, and again this 7 pseudoheptylose 7-phosphates with the help of transaldolase enzyme it will produce fructose 6-phosphates. Then fructose 6-phosphate it will also comes from once again xylulose 5-phosphates with the transcriptolase enzyme, and it will isomerize to form the phosphohexose isomerase, then it will produce glucose 6-phosphates. So in this way pentose phosphate pathway is continuing while glycolysis is hampered or during the process of glycolysis, when if there is any defect, then this pathway is continuing, and so in this way Hexose phosphate monophosphate shunt pathway is continuing. So students, if you found this video helpful, please like the video, subscribe to the channel and turn on the notification bell so you do not miss any biology lessons, and please remember, please comments if any doubt, any queries regarding pentose phosphate pathway, and thank you for watching, and see you in our next class, specifically the significance and other importance factors in pentose phosphate pathway. So